Terumo presents a vision on how all the steps of blood collection and component separation can be streamlined into an integrated process based on technologies that are available today, such as multidirectional data transmission, supported by TOMS, Terumo's middleware data management system, and radio frequency identification. The objectives are full traceability and optimal productivity. A donor arriving at the donation center will present a smart donor card provided with an RFID tag or any other smart ID system. It contains all useful information in compliance with local privacy legislation. Once the donor is identified, the doctor can proceed with the medical interview and checkup. If the donor is considered suitable for donation, the doctor enters the volume of blood to collect, and a unique donation number is created in the blood bank's IT system. These data are transferred to the donor card, the RFID chip of the blood bag, and to the data manager, TOMS, who will in turn send this information to all the available TRAC2. The nurse ID, donor ID and RFID tag of the blood bag are scanned and compared with the information received from TOMS. The TRAC2 then automatically sets the volume and donation can start. The progress of the blood transfer is clearly displayed on the TRAC2 screen. After successful completion, the TRAC2 will send a status update to the RFID tag and to TOMS. TOMS will then forward this information to the central IT system. The filled whole blood kits are stored in an isothermal container. An RFID label with temperature sensor can be included for additional control. The storage boxes can now be sent to the blood processing center. Containers or trays with the taxi blood bags and temperature monitoring RFID tags are registered via an RFID tunnel. The taxi kit IDs, as well as the temperature readings, are processed within seconds. From the RFID tunnel, all these data are transmitted to TOMS and the central IT system. TOMS will then signal to the taxi whole blood network that bags are available for component separation. The taxi kits are fitted into whole blood inserts. The operator ID is scanned and 6 inserts are loaded into the taxi centrifuge. Its lid contains an RFID reader, which identifies the RFID tags of the whole blood kits. If everything's correct, TOMS will allow the separation process to start. At the end of the centrifugation, the integrated presses transfer the plasma and red cell concentrate into satellite bags. When the process is complete, Taxi sends a status update to the TOMS data manager and writes on the kit's RFID tag that plasma, buffy coat and red cell concentrates were obtained. The operator now places the taxi insert into the footprint of the taxi weighing, sealing and labeling station, where the tag data are read by an integrated RFID pad. These data are checked on the TOMS database, and instructions for weighing and sealing are displayed on the touchscreen, while the lab's central IT system creates unique product labels. The operator fits the tubing inside the T-seals, the plasma bag on the left scale, and the red cell bag on the right. Red cell concentrate and plasma are sealed, then weighed. The integrated printer issues the correct labels, which can have a barcode and/or an RFID tag. All data are sent to the TOMS data manager and the central IT system. The central IT system now allocates buffy coats for pooling. The selection data are sent to TOMS and forwarded to the TSCD pooling station. After reading the operator ID, a pooling kit is placed onto the RFID reader for identification. The buffy coat selection is checked via the RFID reader on the left. The platelet storage solution batch number is also registered. If everything is correct, the system allows the sterile connections to be made. Pooling information is sent back to TOMS and added on to the taxi pooling kit's RFID tag. Now, all the buffy coats and the storage solution are transferred to the pooling bag. The empty buffy coat bags are sealed off. The taxi kits are fitted into the taxi platelet inserts. The operator ID is scanned and 6 inserts are loaded into the taxi centrifuge. The integrated RFID reader identifies the RFID tags of the kits. After verification, TOMS sends the process protocol to taxi and centrifugation can start. When centrifugation is complete, the presses transfer the platelets to the storage bag via the ImmuGuard platelet filter. After successful completion of the platelet separation, taxi sends the process data to TOMS and writes on the kit's RFID tag that platelets have been produced. The taxi insert is fitted into the footprint of the weighing, sealing and labeling station. After verification of the kit's RFID data, weighing and sealing is started. The central IT system creates a unique label. All data are sent to the TOMS data manager and central IT system. The end product is platelet concentrate of consistent high quality. As product data are continuously checked and updated at every step of the process, the blood components can be traced all the way back to donation. The lab benefits from increased productivity, and the patient is sure to receive a safe and high-quality product. With this vision of the future, Terumo continues its commitment to provide highly efficient solutions to healthcare professionals and contribute to the well-being of patients.